Hi, my name is Nicole. I'm one of the technicians at PetVets, and I'm going to be walking you guys through a urinalysis today. So I have a urine sample that's been collected on one of our patients in this syringe, and we're going to work on it and get to as many answers as we can. So there's a couple of different steps to the urinalysis, and I have a sheet here that I run through that has a bunch of different components on it. One of them is just looking at the urine to see its color, to see how cloudy it might be, because that can give us a couple of indications as to something that could be going on with the pet. And then we also measure it on this device here, which is called a refractometer. And what that does is it gives us the specific gravity of the urine. So it kind of tells us how concentrated or not concentrated the urine sample is, which is also another key diagnostic indicator of health for this pet. And then the other step is something called the urine dipstick, which has a bunch of little pads here that we're going to put some of the sample on that will get us some results as well. So things like pH, if there's protein in the urine, if there could be white cells present in the urine. Each one of these little colored pads is going to give us another piece of information. And then the last thing I'm going to do is put some of the urine into this tube, and we're going to actually put it in the centrifuge so that we can get all of the cells or any of the things that are in the urine all come to the bottom of this little tube, and then we're going to take that and put it on a microscope slide and look at it under the microscope so you can microscopically see what's going on and look at things like the presence of white cells, the presence of red cells, if there's bacteria, and crystals. So I'm going to run through that process for you guys so you can see a little bit behind the scenes of what it means to do a urinalysis. So I'm going to take the sample. I'm going to take the needle off so I can work with it, because I don't want to scratch the surface of this piece of equipment, the refractometer. So I'm going to take it and I'm just going to carefully drop one little drop on there, and then put the cover over it. And then from here I'm going to look at it into the light. And in here what I'm looking at is a measured scale, and it's going to tell me the urine specific gravity. So what I see here is the line goes to 1.038. So we talk about that as being 1038 if we are trying to have doctor lingo. So that is fairly concentrated. And then I'm just going to clean this off for now so that the urine doesn't dry on there. And then I'm going to take the sample and I'm going to drop it carefully, one drop onto each one of our little colored boxes, pads, and you're going to see some of them change color right away, some of them take a little bit of time to change. It takes about 30 to 60 seconds for us to see the color and for us to be able to look at it and get a reading off of it. So I just let all the color, all of the sample soak in, and then I'm just going to tap off the excess, and then from there I'm just going to take it and compare it. So I just run through, so the first box is glucose, then we're looking at bilirubin, ketone, specific gravity. This one is going to be your blood. So you can see a color change on that one for sure. This box is pH, so that's always a good indicator that we look at, as well as protein. So those are going to be some of the most important ones. So I'm going to look and read the results and then record them on my piece of paper over here. Double check one so I can write it down. Alright, and then to me the sample is kind of dark yellow and it definitely is cloudy. So if you're not sure if it's clear or cloudy, usually what I would do is, you can see the numbers on the syringe. I would turn it around and see, can I see the numbers through the sample? And I really can't. So to me, I would definitely call this sample cloudy. So that's, something to note as well. And the last step here is I'm going to take some of this sample. I'm going to save a little bit in case we have to refer back to it. And I'm just going to put some in the bottom of this tube. By no means does it have to be filled. And then from here, we always run stuff in the centrifuge, and we want to balance it. Otherwise, the centrifuge would actually wobble, wobble, wobble right off the table. So I just have a spare tube over here that just has water in it. And I've labeled this tube with our patient's name so that there can be no confusion. Match them up, they're pretty even. And then from here, I'm gonna put them in the centrifuge, close the lid, set it to our urine sample. So we usually run it either on urine or on blood. And then we spin the sample for about 7 minutes. So from here, once the centrifuge stops, then we'll take that pellet at the bottom of that tube, because it has that cone shape to it. It draws everything to the bottom. That's what the centrifuge does. We'll spin it really quickly, draw all the cells to the bottom so that we can take that concentration of cells and look at it on the microscope. So I'm not gonna keep you for the next 7 minutes, but we'll check back in to see what we see on the rest of the sample in terms of the microscopy. So thank you for watching.